It's 1 o'clock, so. Hi, everyone. I'm Michelle Avila. I work for for CDFW, and this is the lower trophic levels session of the IEP workshop. So if you're here for the phytoplankton and the invertebrates, you're in the right spot. And even if you didn't intend to be here, you're still in the right spot. So we're going to kick off our first presentation with Danny Cox from CDFW, and he's going to talk to you guys about phytoplankton.
Thank you, Michelle. Hello, everyone, and thank you for joining me in my talk. I am excited to open for the lower trophic session with my presentation on phytoplankton and nutrients in restored tidal wetlands. My name is Daniel Cox, and I am the Water Quality and Primary Production Lead at the Fish Restoration Program Effectiveness Monitoring Team at the California Department of Fish and Wildlife. The Fish Restoration Program is tasked with restoring over 8,000 acres of tidal wetland habitat as mandated by the permits for the operation of the State Water Project. The three groups involved with the program include DWR, the CDFW Implementation Team, and the CDFW Effectiveness Monitoring Team. The restoration sites are spread across the North Delta Arc, which spans from the Cache Slough complex down to Grizzly Bay. The effectiveness of tidal wetland restoration can be measured by the following metrics: capacity, opportunity, and realized function. The goal of the Fish Restoration Program is to restore tidal wetlands for Delta smelt, juvenile Chinook salmon, and longfin smelt. At the Effectiveness Monitoring Team, we investigate whether restoration increases the capacity to support listed fish species through improved abiotic habitat and greater availability of food web resources. The Tidal Wetland Project Work Team produced a suite of conceptual models about tidal wetland restoration. And for my talk today, I will focus on these two components of the food web model: nutrients and phytoplankton. I will investigate the base of the food web by testing the following generalized hypotheses developed by the Tidal Wetland Project Work Team. Restored wetlands will have similar phytoplankton biomass as the reference sites, and phytoplankton community composition at restoration sites will resemble communities at their reference sites. The Effectiveness Monitoring Team employs a before-after control impact design for all of its studies that range from water quality, phytoplankton, zooplankton, macrozooplankton, and fish. We compare each restoration site to a reference site that is in close proximity and has a high degree of maturation. We also characterize adjacent water bodies to examine differences inside and just outside of our restoration sites. We began our pre-restoration monitoring in 2017, and the water quality sampling was conducted in March and April. In 2018, we conducted pre-restoration water quality sampling across more months. In late 2018, Yolo Flyway Farms and Decker Island were restored to full tidal action. In 2019, we increased water quality sampling to encompass most months. Restoration was completed at Winter Island and Tule Red in late 2019. Due to the conclusions from our comparison study with other IEP monitoring programs, we changed tactics in 2020. We sampled fewer months, but with more replicates. In 2020, Wings Landing and Lower Yellow Ranch were restored to tidal action, but they will not be included in this presentation. During our visits to each site, we measured water quality parameters using a YSI XO2 sonde. We collected water quality grabs for the analysis of different constituents, and we also collected samples for identification of phytoplankton. The discrete water grab samples were analyzed at Bright Laboratory for chlorophyll A and phaeophytin, which are proxies for algal biomass, ammonium, nitrate, nitrite, and phosphate, which are important macronutrients for carbon fixation by phytoplankton, And we also measured total and dissolved components of organic matter, which are indicative of the detrital food web and will not be discussed in this presentation. Field and lab blanks were submitted weekly to detect potential contamination. To assess our water quality graph data, I checked the data set for the rate of censoring. Censoring occurs when concentrations of an analyte is less than the reporting limit. High rates of censoring hinders our ability to use the analytes as covariates in more robust statistical methods. I then calculated the means and confidence intervals using non-parametric bootstrap methods. Significant differences in chlorophyll A between sites before and after restoration were determined using a two-sample permutation test, since it does not rely on an assumption for the underlying distribution of our non-parametric data. Finally, I tested differences in community composition using only presence-absence data with permutational multivariate analysis of variance and visualized communities using the non-metric multidimensional scaling ordination. I will begin with presenting chlorophyll A results measured from our sondes during site visits in 2019. The data is split up by season, region, and site type. High concentrations of chlorophyll A were observed at Tule Red in the spring of 2019, while all other sites were characterized by lower concentrations. Yolo Flyway Farms did have higher concentrations than its reference site in Liberty Island. In 2020, Tule Red still had high concentrations of chlorophyll A following restoration. However, it was about 1 order of magnitude less than in 2019. Yellow Flyway Farms continued to have higher concentrations of chlorophyll A relative to its reference site. Let's move on to results from our discrete water grab. I would like to first share the rates of censoring in our data across years. Censored and uncensored data are represented by dark and light shaded bars, respectively. In 2017, we only analyzed samples for chlorophyll A and phytophyton. In 2018, we characterized a graded number of constituents and had low rates of censoring. In 2019 and 2020, we had large increases in the rate of censoring for ammonium. This is attributed to the increase of the reporting limit at Bright Lab from 0.01 milligrams per liter to 0.05. Now let's check out the discrete chlorophyll data. Each panel will represent the project named at the top, and the site type within each is named at the bottom. Data from prior to and following restoration is represented by red and blue bars, respectively. Bars inside individual panels that share letters indicate significant differences between them. Error bars indicate the 95% confidence interval. As we saw earlier in our sonde data, chlorophyll A was very high in Tule Red prior to restoration. Concentrations between Tule Red and its reference site following restoration were not significantly different. At Winter Island, we see low concentrations across site types and restoration status. We did, however, observe significantly higher concentrations at Winter Island than its reference site following restoration. Low concentrations of chlorophyll A were observed across the board at Decker Island. At Yolo Flyway Farms, we did detect significantly greater concentration of chlorophyll A relative to Liberty Island following restoration. Moving on to ammonium concentrations. We see some significant differences across site types in Decker and Yolo Flyway Farms projects. For nitrate and nitrite, We observed variability across projects, but no significant differences. And finally, for phosphate, we observed significant differences between Decker Island and Horseshoe Bend. However, the concentrations are very low. Now I would like to share some results of the algal communities at the tidal wetlands. I will begin with the comparison of the data produced by two different contractors. Circles will indicate samples processed by BSA, which we have contracted since March of 2020. Triangles represent samples from 2017 to 2020 that were processed by eco-analysts. Colors represent the different regions where the samples were collected. You will also see ellipses, which indicate 95% confidence intervals for each group. As you can see, there was very little similarity between samples processed by the two contractors. About 24% of the variability could be explained by contractor differences alone. Now I will overlay the species names on the same ordination. The species in red are unique to BSA and species in blue are unique to eco-analysts. The species identified by both contractors are in green. As you can see, the differences in algal community composition are likely driven by methodological differences between contractors. Due to this discrepancy, I will now show an ordination of data produced only by eco-analysts. Regions and site types are represented by color and shape respectively. The overlap in ellipses for each region indicates a degree of similarity and community composition from each region. Results from the Permanova show that region and the interaction of region and site type explain about a quarter of the variation in community composition. Finally, I subset the data further to assess similarity in composition at Decker project prior to and following restoration. This site was the only site with enough data from eco-analysts to perform this analysis without including the effect of contractor. Points and ellipses for restoration status are represented by shapes and line types respectively, and site type is indicated by color. There was a high degree of variability in composition prior to restoration, followed by a greater degree of similarity across site types following restoration. The interaction of year and season actually explained a greater proportion of the variation in community composition than did restoration status. With all of that, I would like to move on to a brief discussion. Biomass as measured by chlorophyll A did show similar results between SOND and GRAB data. The restored wetlands were characterized by having similar or even greater chlorophyll A than the reference sites following restoration. Even though pre-restoration data at some sites revealed high concentrations of chlorophyll A relative to post-restoration observations, it does not suggest that restoring sites to tidal action was a failure. The act of restoration improves the opportunity for listed species, listed fish, to access the abiotic habitat and food resources within the wetland. It also improves the exchange of resources to adjacent water bodies that are characterized with low availability of food. Finally, biomass is just a snapshot of the standing stock of algal resources. Ideally, we would like to incorporate measures of production and flux to better understand the contributions of high-quality carbon sources by algal assemblages to restored wetlands and their surroundings. As for community composition, we did find that Decker Island had similar algal constituents following restoration. However, we need more observations across water year types to assess contributions of flow to the variability in composition. I would now like to raise some of the challenges we need to overcome. The increase in the reporting limit has taken a toll in the world of ecological monitoring. Here, I would like to share a time series of ammonium data from the Environmental Monitoring Program. The red line indicates a reporting limit of 0.05 milligrams per liter. As you see, EMP had many observations below 0.05 milligrams per liter when the reporting limit was set to 0.01. Similar to our study, EMP data shows a clear time step difference in the reported ammonium concentrations. Unfortunately, this increase in the reporting limit coincided around the same time that the regional sand wastewater treatment plant underwent an upgrade to drastically reduce its ammonium loading to the system. Ideally, the reporting limit should be lowered so that we may detect ecologically significant ammonium values that exist lower than 0.05 milligrams per liter or 3.5 micromolar. Lastly, it is important that we find a way to handle artifacts and community composition data sets that are produced by contractor methodologies. Fortunately, a new phytoplankton project work team has been developed, and we can work together to address this issue. And with that, I'd like to thank our funders, DWR and the state water contractors, and I would like to thank you for attending my presentation. I will now open it up for any questions.
All right, thank you, Danny, for that presentation. If anyone has any questions, feel free to raise your hand or just submit them in the Q&A question box, and we will get to them if we can. Right now, there's no questions in the box, so we can also sit here awkwardly for a couple minutes. So if you have any questions, submit them. All right, we have one question. Can we just switch labs for ammonia?
That is one option. We would need to work with how we can fund a different lab to produce those results. At this moment, though, that's not something we can do easily.
All right, Peggy Lehman asks, when you looked at the diatoms only, was there an increase in biomass for restoration projects?
That's an interesting question. I have yet to delve into specific groups alone and into the biomass data. We're working with a few issues that were presented by BSA recently, but that will be one of our next steps.
Okay, Jenna says, thanks, Danny, for a great talk. Let's definitely chat in the PWT.
Definitely.
Rosie wants to know, what was the biggest member of the phytoplankton community?
In the BSA data set, it was mostly dominated by Eucapsis, and the eco-analyst data set did not report any hits of Eucapsis, so that's one of the things I'd like to get to the bottom of, whether they were identifying the same thing differently or not. But we'll have to look across other data sets that have used the two contractors as well and see what we can do next.
Okay, I think we can squeeze in one more question. Ted Flynn asks, is it possible that the biomass was lower because the consumption rate by zooplankton was higher?
That is a good question. It could be that reason. It could also be that residence time is lower due to tidal action that has been restored. But we gladly welcome any studies looking into that.
All right, thanks for that, Danny. Good talk. Our next presenter is Michelle Jungbluth from San Francisco State, and she's going to be discussing copepods.
All right, can you see the right screen?
It's still in presenter mode.
Oh, okay, I'll swap. All right, is that better?
Yep, thanks.
Fabulous. Thank you very much for being here today. You might notice my title is a little different. This is very fresh data, so the topic slightly changed, but it's all, you know, about lower trophic levels. So I'll be talking about the dietary DNA analysis of Copepod diets and environmental DNA in the northern San Francisco estuary. This is work that I've been doing in collaboration with folks from mainly the Kimmerer lab and Slaughter and Tony Ignofo. So here, of course, we're all well aware in our San Francisco estuary, the native and introduced fishes are declining, and that's from many complex and interacting factors. One of the most important factors, I think, is that introduced species have changed the food web. One major introduction, of course, was that of Potamocorbula amurensis, which resulted in a decline of chlorophyll across a range of salinities after its introduction, and chlorophyll is a commonly used indicator of food for zooplankton. In addition to that, there were additional introductions in fish food abundance type and biomass in our copepods and mysids, primarily things like differences in species composition, an introduction of the small Limnaithona, which tends to be very abundant, but it's a small species, and Pseudodioptimus forbesi, in addition to changes in the mysid composition. And so, as I mentioned, chlorophyll is commonly used as an indicator of food for our zooplankton, but it's becoming more and more clear that in our estuary, chlorophyll is not always a good indicator of food. A recent study that came out by Geardy et al. From the Kimmerer lab found a sort of meta-analysis of data that they've collected, the Kimmerer labs collected over the years, looking at growth and egg production rates compared to chlorophyll levels. And there was a very weak relationship between growth and egg production rates and, in relation to chlorophyll. In addition to that, even during blooms of phytoplankton, I found in a study we did looking at ingestion of the primary bloom organism that the actual ingestion rates were quite low and unrelated to egg production. So it seems that chlorophyll is just not a great indicator of food for our copepods. And this has also been seen in other estuaries like Chesapeake Bay. A similar observation was made with Copepod carchatansa, where egg production was more related to protistan biomass, not phytoplankton and temperature of that bay. So this kind of has raised many questions. What are our copepods actually consuming, and are there types of prey that are indicators of good food for primary consumers? So these are questions that we're focusing on in our lab right now. Now, there has been one, well, there've been a few prior studies, but one using dietary DNA analysis is shown here by Ann Holmes, now a PhD student at UC Davis. She described with high throughput sequencing the 16S rRNA gene, the patterns of feeding in a cache slough complex of Pseudodioptimus forbesi on photosynthetic prey, and found a high sequence abundance of cyanobacteria, which is thought of as a poor food in the diets of this species. Especially, it was higher compared to the apparent availability in the water column as shown by sequencing, and a low sequence abundance of cryptophytes, which are thought of as good food, which were much more abundant in the water. So that triggered some additional questions. Is the actual ingestion of cyanobacteria high? And that question we are starting to tackle. A student at SFSU, Cheryl Patel, designed and applied a qPCR assay to measure cyanobacteria ingestion. Those results are in progress. So that's a little teaser. Hopefully in the next year, we'll be able to get those results out, and we'll see whether direct measurement of ingestion shows the same patterns. But what I'm going to talk about today is 2 recent studies of food web support across lower salinity habitats in the estuary. Our first study is called Hunger Games, which you may notice there's a talk later today with the same name, but it's a different study. We just happen to have a common fun name. We even designed a logo right here. So this Hunger Games study was from 2019 to 2021. The samples collected and analyzed here were in 2019, where we did a range of measurements to look at feeding by our copepods on field collected samples in situ. We did dietary DNA analysis and water sample analysis with high throughput sequencing of the 18S and 16S genes. And the 18S is good for characterizing eukaryotic prey, while the 16S is better for prokaryotes and bacteria, primarily bacteria. We also did other measurements to infer feeding, like microscopy, chlorophyll, and flow cytometry. Other things that we did that I won't talk about are feeding incubations and growth and egg production rates. But that'll be for a later talk. The second study was the Sacramento River Nutrient Change Study, aka we call it the son of baseline. There was a baseline study. This is a sort of second baseline study to look at the changes in response to the wastewater treatment plant reducing the amount of ammonium loading. But I won't be connecting my data to that yet today. That's in progress because all of this data came hot off the sequencer in December. So it's only been in my hands for a few months, still kind of working through the fun details. But anyway, so for this one, also we did in situ feeding of the 18S and 16S, dietary DNA and water, also chlorophyll, growth and egg production, and have other data for environmental context. So for methods, I did DNA metabarcoding, where I cleaned and extracted sets of 5 adult female Pseudodiaptomys forbesi, and we collected water samples that were pre-filtered to remove large organisms and filtered onto a 0.2 micron Cervex filter. These were then amplified with the targeting both 18S V7 region and 16S gene targets to characterize again the eukaryotic and prokaryotic assemblages. Then sequenced with aluminum-IC metabarcoding for metabolomics. And then the data that we got from that, I applied a newer method of classification in QC called the ANACAPA toolkit. This is developed by California eDNA program. And I applied that mainly to the 18S gene because there were some challenges with 16S I won't get into here. But with the 18S sequences, we used the primers to create a custom reference database. We then used data2 in this toolkit to do quality control and assignment of ASVs, the 100% similar OTUs, sequence groups basically. And then you classified the sequences against this reference database that was developed using primers. With the 16S gene, we did use the same data2 for QC and ASV assignment, and then classified the sequences using Mother and the Silva database. So as for results for the 18S gene, just to give an idea of it, broad measure of eukaryotic sequence diversity across both studies. I did a PCOA with Bray-Curtis dissimilarity index just to show how the diversity within each sample compares to each other. and across different types of samples. And you can see that the water samples clustered tightly together as far as their community similarity, that's in blue and yellow. And the copepod samples were grouped but spread a bit more, more depending on where and when they seem to be collected and feeding. But we do see groupings by broadly sort of steady area. For the 18S gene for the Hunger Games study, so that's the eukaryotic prey, the main prey that we found in the Copepod diet were green algae, goodness, chlorophytes that are green algae, kinetoplastia, a type of flagellate, and Saccharomycetes, which is a fungus. Also, a parasitic alveolate was abundant. And I'm just mostly describing the groups that were common across most of the Copepod diet samples. And then those that were more abundant across the water samples were the group Cocinodiscophyceae, a group of diatoms, cryptophytes, Dinophyceae, which is a group of dinoflagellates, and some diverse others at lower relative abundance. When we look at the 18S gene for the son of baseline study, there are more samples from that study, and interestingly, some similarities and differences. We see some chlorophytes in the Copepod diets, which are the green algae, a sponge, which I have to validate, a ciliate group, Oligohymenophora, green algae, Zygnemophyceae, and an unclassified group that were common in the Copepod diet. In the water samples, the similar Cocinodiscophyceae diatoms, cryptophytes, some ciliates that were in the group Spirotrichia, some bivalves, interestingly, in the water samples, and some diverse others at lower relative abundance. Now you can see if you look at these percents of sequences, there's a huge diversity of things that were at lower relative abundance. So there are a lot of things in these samples, just haven't been able to pick up the patterns yet, other than the dominant prey. So in the 16S, a similar pattern is shown with the PCOA of the diversity in each sample, clustering of the water, and sort of general grouping of the Copepod diet samples, although they're a bit more spread out, which is kind of interesting. When we look at the actual diversity in those samples in the Hunger Games study, we can see, again, this is in the prokaryotic prey with the 16S gene. In both types of samples, there were a couple of groups that were common. Alpha proteobacteria and cyanobacteria were common across both, and those are common water bacteria. In the Copepod diet, there were quite a few bacteroidea and gamma proteobacteria, which are likely a microbiome signal, actually. So that's one thing that I had to do, is sort of pick apart and see which ones are likely microbiome versus actual prey. And then in the water samples, there's Acidomicrobia and Actinobacteria, which are common soil-dwelling microbes, and Chloroflexia, filamentous green non-sulfur bacteria. In the son of baseline study, or the 16S prokaryotic prey, in both samples, we found Bacteroidea, which is a common microbiome organism in both types of samples, actually, and alpha proteobacteria and cyanobacteria, again, the common water microbes. And the Copepod diets, some bacilli, which is an interesting extremophile, that's spore-forming bacteria, and gamma proteobacteria, which is likely a microbiome signal, and then a group of unclassified bacteria, so I have to dig into that one. And then in the water samples, Acidomicrobia and Actinobacteria, which are those soil-dwelling microbes again, and a group of aquatic plant-associated bacteria, which are called Armatomonadea. That's fun. So yeah, lots of fun scientific names. So in summary, across multiple studies of Pseudodiaptans forbesi, dietary DNA, and water samples, we found in the prokaryotes the most common and abundant taxa, sort of summarizing across both studies. In the diets, we found alpha proteobacteria, Bacteroidea, gamma proteobacteria, and cyanobacteria. Some of these are possibly microbiome signals, others are actual diet items. Some of those microbiome organisms were also in the water, which is kind of interesting. So might have to come down to looking at finer levels of taxonomy, which I haven't gotten to do yet, but that will be interesting. In the water samples, yeah, again, Amphipod bacteria, Acidomicrobia, and cyanobacteria. So those were both in the diets and water. In the 18S eukaryotes, those seem to be more variable across the different studies, but the common and most abundant taxa were chlorophytes in the diets, and in the water, diatoms and cryptophytes. So that sort of suggests that maybe the eukaryotic assemblage may be more sensitive to regional variations in conditions or to predator assemblages than prokaryotes, but that's just a wild guess at this point. I did find some interesting evidence of pathogens and parasites in many of the copepod samples, so we might look into that a little bit more. As far as next steps, again, this is hot off the press, so having over 20 million DNA sequences to sort of classify and figure out what's real and what's not real. You know, this is all very preliminary, but it's very exciting and fun. I still have to do the science, so I'll be connecting the dietary assemblages to environmental conditions, growth rates, reproductive rates, comparing this dietary DNA to other measures of ingestion from bottle incubation, specifically for the Hunger Games study, and assessing for impacts of changes in the nutrients in that Sun baseline study on dietary or health indicators. And I'd like to thank a lot of people shown here. And if there's, yeah, I think there might be time for questions. Happy to take your questions.
All right, Michelle, thanks for the presentation. You have a question from Peggy Lehman. It says, the diet was high in chlorophytes. Your picture shows volvox for a chlorophyte, or the zooplankton eating volvox. These are very large organisms.
I guess they didn't show up as a large component. I would have to look at, you know, there's, for my overall summary, I just kind of looked at, you know, the big groups, but I believe they showed up in at least some of the diets. So I'd have to dig in a little bit more to see if volvox was there. Yeah.
Arthur has his hand raised. So Arthur, if you want to go ahead and ask your question, go for it.
Yes, thank you. Is there a way to calculate selectivity using these genetic methods?
It's a little bit tricky because the high throughput sequencing isn't precisely quantitative. You get relative abundances, but you can do something similar. So in gene expression studies, which are sort of similar, you can look at sort of the relative comparisons between, you know, within a subset of taxa, comparing what was in the environment to what was in the diet. I did something similar in the longfin diet paper that I did to sort of hint at whether or not the selectivity was there, but it's tricky because it's not quite quantitative. Yeah.
Okay, Christina Birdie would like to know, do you think that other herbivorous calanoids like Eurytemera or Cynocalanus have similar diets given their similar ecologies?
That's a good question. I'm sure there's some overlap, but it would take a little bit of sleuthing to figure out, you know, do their mouth parts differ with, you know, whether they're more, I don't think Cynocalanus is predatory. I'm not sure, but we do have a student, Amy Wong, who is comparing diets in Eurytemera to Pseudodioptimus. So in the next couple of years, we'll have some exciting work looking at those two species at least with high throughput sequencing. I imagine there's probably some overlap, but there would likely be differences.
All right, we have another question. I'm sorry, I'm not going to say your name because I'm sure I'll butcher it, but it says, could the diet composition differ in different habitat types?
Yeah, and I think that I think that my results sort of hint at that, but it's not shown super clearly yet. I think the PCOA that I showed it sort of separating out in the Copepod diets, even though the water samples were more similar to each other. The Copepod diets really spread out. So I think, yeah, I think they do, at least on some level. I think there will be differences.
Okay, unfortunately, we don't have time for any more questions. So if you have more questions for Michelle, I guess send them her way. Our next speaker is Amanda Ray McCormick from UC Davis, who is also going to be talking about copepods. So, yep, there you go.
Hello, my name is Amanda McCormick. I'll be talking about the effects of wetland plant material on copepod survival, growth, and resource selectivity. I would like to start by thanking our funding source, the California Department of Fish and Wildlife, as well as my numerous co-authors and collaborators on this work. I'd also like to thank multiple individuals who have assisted both in the field and lab for these experiments that I'll present. I'd like to provide a bit of context for the Delta's lower food web. Of course, the Delta has undergone many major ecosystem changes and stressors. These include over a century of dramatic land use alteration, substantial water diversions, and numerous invasive species introductions and establishments, in addition to multiple other stressors. These have culminated in depressed pelagic primary production in the Delta. And what this means for many pelagic primary consumers, including copepods, is that they're chronically food limited. This, of course, affects the entire pelagic food web, as these primary consumers are an important trophic link, such that documented pelagic organism decline across the entire food web has been seen. In the Delta, wetlands play an important contribution in food webs. Both historically, they were considered a key contributor of bioavailable organic matter to the Delta food web, and it's one that's been severely impacted by land use change. For example, there's been a dramatic reduction in wetland area. So you can see in this figure from Clarn et al. That historically, wetland plants in the green contributed a lot to primary production, and with those declines in area, that contribution has become smaller. But nonetheless, wetlands, there's reason to believe that they contribute important resources for the lower food web. As one example, floodplain wetlands have been shown to support high zooplankton densities and fish growth rates. And then in work that's closely related to what I'll be presenting on today, a study led by Jen Harfman showed that copepods actively selectively consume wetland plant material, and that this material can increase their survival. And this is in spite of the fact that wetland and plant detritus is typically considered a lower quality food resource than others, such as phytoplankton. And so what this previous work suggests is that within ecosystems in which primary production is chronically low, such as the Delta, these supplementations from wetland may be important for the food web. And the objective that I've been thinking about when doing this work is wanting to understand the context in which wetland detritus may benefit copepods. And specifically, I've been thinking about whether the influence of wetland detritus on copepod survival and growth is dependent upon the availability of other resources. And specifically, I've been thinking about phytoplankton. And I've been exploring this kind of context dependence of detrital effects through a series of experiments. And I'll be talking about first results from fieldwater experiments where we're measuring copepod survival, growth, and resource selectivity, and then a smaller set of results from some manipulated diet experiments. I'll first start by talking about our field water experiments. So from the Delta, we collected water from 2 regions. Our choice of sites was in part motivated by wanting to capture a variety of resource composition that would be available to copepods. So we selected sites from 2 regions. One is Grizzly Bay. This is a higher salinity area. It's in relatively close proximity to a very large intact wetland in Sassoon Marsh. And then our other sites were in the northern part of Liberty Island. So these are quite different from Grizzly Bay. The salinity is much lower, nearly freshwater. Within each region, we had sites in 2 different habitats. So we aimed to choose sites that were relatively shallow water or open water habitats. And again, the motivation was in trying to create contrasts, potentially in terms of the resource composition available at our sampling locations. And we collected water 3 different times with corresponding experiments that were set up. And from the field water, we used a couple biomarkers to assess the resource composition and availability. So we used chlorophyll kind of as a proxy for phytoplankton and lignin as a biomarker for vascular plants. The design of the experiment crossed our 2 regions, Liberty Island and Grizzly Bay, with the shallow and open habitats, and we also imposed 2 diet treatments in the lab. The first was no food, so this basically meant just the field water as we collected from the field, and then a food treatment, which was the daily addition of algal food from the lab. We also set up control treatments, which included reconstituted water plus the same algal food as our food treatment, and we had different controls for each region with the corresponding salinity. We used for our experiment juvenile Eurytemera ephinus from a laboratory culture. The densities were 10 copepods per liter, and we conducted 4-day experiments. I'll first start by briefly showing you what we found in terms of biomarkers at our different sites. So I'm showing both chlorophyll as our proxy for phytoplankton and lignin as our proxy for vascular plant material. And what we saw is that for chlorophyll, our concentrations were pretty low to moderate with a high of about 7 to 8 micrograms per liter. And across experimental rounds, kind of the site that had the highest versus lowest chlorophyll showed some variation. So you can see, Liberty Island Open was the highest in September, but not necessarily in the other collection dates. In contrast, lignin showed more consistent differences, at least from region to region, with Grizzly Bay being higher than Liberty Island. After 4 days, we assessed survival of the copepods in our experimental beakers. So what I'm showing is for the 3 rounds of experiment from both Grizzly Bay, Liberty Island at the different habitats. And what we found is that for all round region combinations, survival was generally high. And likely this is in part because the experiment only lasted 4 days. So potentially there wasn't enough time for any substantial survival effects to take place. At the end of 4 days, we also measured body length of all surviving copepods. And so what I'm showing here is a figure set up in the same way as the survival one. And I'm showing growth in terms of the mean body length from each beaker compared to the mean initial body length of copepods that were stocked into the beakers. I'm going to go through a few things that I'd like to highlight. So the first is that the addition of algal food, unsurprisingly, positively affected copepod growth. So if you look at the food treatments compared to the no food treatments for all of the different region and experimental round combinations, adding algal food increased the growth. In general, what we found as a second kind of takeaway is that when you look at the field water with the supplementation of algae, growth rates tended to be higher than controls that received that same algal food. And what that suggests is that there's likely resources in the field water that are important in contributing to growth. And perhaps what, at least in my opinion, is the most interesting about the growth results is that we saw variation in growth rates, in particular in the field water without food, and variation in growth that may be linked to resource composition and availability. So I've highlighted a couple examples here that I'll go through. So the first we saw in Grizzly Bay at the shallow site from September, we saw that growth when we just gave copepods field water, no algal addition, it was actually matching the control treatments, which are replicating kind of comfortable lab conditions that the culture is normally experiencing. And if we think about what our biomarkers tell us about this site, we see that the site had kind of moderate, not super high chlorophyll, but also high lignin. In contrast, if we examine both habitats from Liberty Island in April, we see that growth was quite low in the beakers without algal food supplementation. And even when we added algae, we were still seeing growth rates that were basically only equivalent to what the control beakers were receiving. And so what this suggests is that in the field water itself, there may not have been that many resources that were contributing to growth. And if we look at what those biomarkers match to, we see that both chlorophyll and lignin were quite low. And by examining all the combinations of our biomarker data along with our growth data, we can start thinking about how variation in growth might be linked to the resources available. So one thing that we see when examining our growth rates is that the highest growth, which are the darkest blue dots, seem that they might be associated with at least moderate chlorophyll levels and also relatively high lignin. But what I'll caution is that our biomarker data provides only a really coarse assessment of resource composition and availability. So for example, even at our highest observed chlorophyll levels, we didn't see particularly high copepod growth, but we also don't know, for example, what these algal taxa were and whether they're palatable or nutritious for copepods. So as a next step, we currently have amplicon metagenomic samples being analyzed, both from field water and copepods. And what this will allow us to do is assess resource selectivity of copepods and potentially find specific resources to the species level that are positively associated with copepod growth. I'm next going to show a smaller set of results from some manipulated diet experiments, which I'm still working on. So again, a large part of my motivation in this topic has to do with what is the context under which wetland plant material may benefit copepod survival and growth. And so in these experiments, I've been examining the effect of wetland detritus across a gradient of algal quantities. And the algae I've been using is chlorella from a lab culture. The wetland detritus material I've been using is tule. So this is a common wetland plant throughout the delta. And so I collected senescent material from the field and it was dried and ground to a small size fraction. So I've been crossing the 4 chlorella quantities with 2 levels of tule, either control or not receiving tule, and an addition of tule. I've been conducting these experiments again with juvenile Eurythemera that are from a laboratory culture. I've been doing these experiments in 12 well plates where each well contains a single copepod. I'll show results from 2 experiments. One was designed to assess survival time, and this lasted over 50 days. Essentially, it was run until there were no longer any surviving copepods. The second experiment was designed to assess copepod growth. This was done over the course of a week. So I'll first show results from the first experiment. What is shown on the y-axis is the number of days survived by each individual copepod. The x-axis is showing you that gradient in chlorella quantity, Individuals receiving tule are shown in red. Those that did not receive tule are in gray. So what we saw is that the higher chlorella quantity increased individual survivor time, but I did not see an effect of tule nor an interaction. I will say that one potential complication in this experiment is discerning the effects of diet versus the natural life cycle on mortality of the copepods. So specifically around day 30, I started to see fairly dramatic mortality incidents, which likely coincides with the natural life cycle. So I'm rerunning survival experiments with assessing survival at different kind of endpoints rather than letting them run until there are no survivors. In the growth experiment at the end of 7 days, I'm showing you the body length that we saw in copepods, again, across the chlorella gradient, and tule in red, gray for no tule. We saw that chlorella quantity increased copepod body size. The effects of tule are a little bit more complicated and uncertain potentially. So if you look at the full model, which contained a chlorella and tule main effects as well as their interaction, we don't see any significant effect of tule. If you look at the effects without considering the interaction, what we see is there's a marginal, so not statistically significant effect of tule, but a positive effect on body size. Although this effect is very small, and again, it's only marginal if you don't consider the interaction. So more information I hope to learn from this experiment is examining selectivity of chlorella and tule using a qPCR assay to see actually if they're actively ingesting one resource compared to the other at a higher rate. And also what I found is growth is somewhat stunted in these plates, and so I'd like to run a second growth experiment using a somewhat different kind of design in terms of experimental unit. So I'd like to finish with a few conclusions. First, from our fieldwater experiments, they suggest that vascular plant material in combination with phytoplankton may be promoting copepod growth. And from our future metagenomics results, we hope that will provide a way to directly assess resources that are positively associated with these growth rates. Our diet manipulation experiments do not yet provide a clear picture of wetland detritus effects on copepods, but these experiments are still underway and I'm still carrying them out. So overall, what I would say is wetland detritus may in part support copepods in the presence of other resources such as phytoplankton. And with that, thank you for listening, and if you are interested in these topics, please feel free to get in touch with me. Thanks.
Okay, it looks like we have a few questions in the chat. Wim said, Amanda, I may have missed it, but was the particulate matter kept in suspension during the experiments? This is very important because most of the food taxa will sink to the bottom.
Yeah, that is a good question, and I had not thought about the implications, but no. So in the beakers, they were bubbled kind of gently, so potentially that provides some kind of mixing, but you definitely could see things settling on the bottom. So I do think that some of that particulate organic matter was falling to the bottom of the beakers, and that is a good point that would potentially mean that those resources weren't necessarily available to the copepods in the experiment. I do have field copepods that I hope to compare the metagenomic analysis of the copepods from the field and water from those same sites. So I'm hoping that would potentially alleviate that if that was a concern for looking at diets here.
Okay, have you looked at the impact on growth and survival of epiphytic periphyton growing on wetland detritus?
Yeah, I think that's a good question. I think I should have maybe been clearer when the detritus I've been considering, I guess, is like particulate organic matter that is in the water column already from the delta. So it's a little bit different than something that's in things that are kind of like at the 0.45 or at the 45 micron size. So I'm not sure if the pieces I've been using would be large enough to support such epiphytic periphyton, as opposed to, you know, if you have like larger detrital pieces, more like coarse particulates.
Okay, thank you for your talk. Unfortunately, we don't have time for any more questions. So if you're interested, I guess contact Amanda directly. Our next speaker is Arthur Barros from CDFW.
Hi, everyone. I'm Arthur. I'm an environmental scientist working on the zooplankton study, and today I'm going to be discussing the responses of zooplankton communities in the San Francisco estuary to drought. I'll first briefly cover the zooplankton results of the drought synthesis report, and then show you some expanded analysis that goes beyond that report. Starting last year, the IEP drought mass was prompted by yet another record-breaking drought to take a more holistic approach at analyzing major ecosystem responses to drought within the estuary and the delta. This group was led by Rosemary Hartman at DWR, and you can see her talk going over the results of that drought synthesis report during session 3. And for this report, we utilized many different long-term data sets to analyze the drought impact on things like flow, water quality, nutrient availability, phytoplankton, zooplankton, aquatic vegetation, and fish. We also attempted to standardize our analysis and modeling for all these environmental parameters. So for the zooplankton analysis, we looked at the following metrics. First, we looked at we utilized metrics of BPUE, or biomass per unit of effort, measured in terms of cubic meters of water sampled. And then also, due to California's high interannual variation in precipitation, as well as its well-developed water infrastructure systems, a single dry year does not necessarily produce a drought. So here we designated years as either drought years, being 2 or more consecutive years with an SVI below normal, SVI being a Sacramento Valley index, or as wet years with 2 or more consecutive years with an SVI above normal. Other years were considered neutral, and you can see how that plays out in the figure at the bottom of this slide. We also wanted to make sure we were examining regional responses to drought, so we divided the delta into 5 regions: Sassoon Bay, Sassoon Marsh, the Confluence, the South Central Delta, and the North Delta. And due to the low amounts of long-term monitoring in the North Delta, that region was not included for our zooplankton analysis. And I wanted to briefly mention how zooplankton sampling is done for the environmental monitoring program, because that's the data that was mostly used for this analysis. So we used 3 different gear types to collect 3 different size classes of pelagic zooplankton. We used a Mysid net for macrozooplankton, a CB net for mesozooplankton, and a pump for microzooplankton. And the Mysid and CB net are towed in a stepwise fashion behind our vessel, and the pump is raised and lowered through the water column at a stationary point. If you want more information, you can check either of these 2 links. We'll briefly look at some of the zooplankton results we looked at in the report. First off, looking at overall zooplankton BPUE across the whole delta, we didn't actually see any significant differences between drought and wet years when looking at the entire region as a whole. When we break up the analysis by season, we start to see some significant interactions, with wet springs having the lowest zooplankton BPUE in the delta, and wet or neutral summers having some of the highest measurements that we've seen. When we include region in this analysis, we can see that with both Sassoon Bay and the South Central Delta, drought do have significant impacts on BPUE. With Sassoon Bay, we see a decrease in BPUE during drought years, and then in the South Central Delta, we actually see an increase in BPUE during drought years. We also looked at some short-term results to include a wider data set. So here we saw that, just looking at the data from 2010 to 2020, we saw that water year, region, and month all had significant impacts on zooplankton biomass. Then finally, for the report, we looked at overall delta zooplankton community differences between drought, wet, and neutral years. Here we used non-metric multidimensional scaling ordination to describe the variance between the communities. And using permutational analysis of variation, we saw that about 26% of community variation could be attributed to drought conditions. But so the analysis we present in the drought mass report is a fairly broad look at zooplankton density changes in the delta in response to drought, but we wanted to expand upon that for further analysis, because when we look at the regional communities and species-specific interactions, we start to see some really interesting results that we can tease out. So next, I'm examining taxa-specific data using the EMP data from 1994 to 2020. The reason I start at 1994 is because not all the taxa we looked at in this analysis were established in the region before that time. So I'm going to be bringing this figure up again several times throughout this talk, and this comes from the original report. This breaks up the impact of drought on BPUE by region. So looking back at this figure, we're going to look first at Suisun Bay, and we can see in Suisun Bay that overall biomass did decrease during drought years, which didn't really occur in any other regions. But then we also want to break up the BPUE differences between different taxa. Here we can see some different interactions between these different species. And of course, I can't talk about all the taxa because time is limited. So I'm going to highlight a few key species from each region and describe their response to drought. So first off, we can look at Pseudodiaptomus forbesi, which is a very important calanoid copepod food item for pelagic fish in the delta. And we see that in drought years, its BPUE significantly lowers in Suisun Bay. We see that same pattern with the other important calanoid copepod, Eurythemera affinis, which also decreases significantly during drought years. However, if we look at the small and the invasive copepod Limnoithona tetraspina, we see no significant effect from drought on their population within the Suisun Bay area. Then we can look at the native mysid, Neomysis mercedes, which is pictured at the bottom of this picture, and compare it to the non-native mysid, Hyperacanthomysis longirostris, at the top. You can see that the native mysid and mercedes actually does have a significant decrease in BPUE during drought years, whereas the invasive H. longirostris sees no impact during drought years. And finally, for this region, we can look at Cladocera, which are another important source of food to a lot of the pelagic fish in the estuary. And we see that in Suisun Bay, both Bosmina longirostris and Daphnia see substantial declines during drought years. So overall in Suisun Bay, we mostly see decreases of very important zooplankton during drought years, with the smaller invasive species seeing no real significant effect. Next, we're going to look at Suisun Marsh. So calling back this plot, you see here Suisun Marsh, we don't see any significant difference in overall zooplankton BPUE between wet and drought years. However, looking at individual taxa responses, we start to see some different patterns. So again, we're going to look first at the copepod Pseudodiaptomus forbesi, and we see the same pattern that we saw in Suisun Bay, where we see a decrease in their densities during drought years. But if we look at the other calanoid copepod Eurythemera affinis that we saw in Suisun Bay, they decreased in density. Here in the marsh, we don't see a significant impact during drought years. Looking back at the mysids, we see a similar pattern to Suisun Bay, where the native Neomysis mercedes does significantly decline during drought years, but again, the invasive H. longirostris doesn't have any significant impact during drought years. And then again, with we can look at the Cladocera, and they also have the same relationship as they did in Suisun Bay, with both of the Cladoceran species taking a significant hit during drought years and seeing significant declines. But we can look at the invasive, and again, very small, Limnoithona tetraspina, and we actually saw a different pattern. We see substantial increases in density in the marsh during drought years, which we didn't see amongst any of the other taxa we saw in that region during drought years. So looking at the Suisun Marsh region, we see several important species, again, decreasing, but we also see an increase in the tiny and invasive L. tetraspina. Next, we're going to look at the confluence, and similarly to Suisun Marsh, when you're looking at the region as a whole, the confluence does not see a significant impact of drought on the overall zooplankton BPUE. But again, when we look at taxa-specific responses, we see a little bit of variety. We're going to look at Pseudodiaptomus forbesi again, And here, though, the impact of drought on densities of P. forbesi has become basically non-significant. And this basically carries over to most of the other taxa we were talking about. So I'm not going to cover them in as much detail, but basically throughout the confluence, most of the taxa did not see a significant difference between drought and wet years in terms of densities. However, when we again look at the invasive Limnoithona tetraspina, we see that in drought years, again, just like they did in Sassoon Marsh, their densities actually increase in this region. So overall in the confluence, we don't actually see a lot of change during drought years in terms of their densities, other than an increase in the invasive L tetraspina. For our final region, we're going to go ahead and look at the South Central Delta. And here, unlike the other regions, we can see there's actually a significant increase in zooplankton BPUE during drought years. So this didn't happen in any other region, so pretty surprising. Again, we can look at specific taxa to see what is causing that overall BPUE increase. So my first thought was L tetraspina, it's increased in the other regions during drought years, very likely increasing in this region, but that's not the case. In the South Central Delta, we don't see significant changes to L tetraspina's densities between drought and wet years. And that holds the same for Pseudodaphnis forbesi, the important calanoid copepod. No significant differences in densities between wet and dry years in the South Central Delta region. However, what was very interesting is the other important calanoid copepod, Eurytemera affinis, does see significant differences between the drought and the wet years, with actually higher densities during drought years, which is not something we would have expected to see. But if we go back and look at our Cladocera, our two main Cladocera, Bosmina and Daphnia, we actually did not see a significant impact on their densities during drought years. And finally, I wanted to bring up a slightly different taxa, Diaphanosoma, which is another important Cladocera that we mostly see in this region. And here in South Central Delta, we see that Diaphanosoma also increases in BPUE during drought years. So its density goes up during drought. So overall in the South Central Delta, many species actually see increases in BPUE during drought, and none of the taxa we examined saw decreases during drought years within this region. So to wrap things up, we can kind of look at the entire region, where we see that in downstream areas such as Sassoon Marsh and Sassoon Bay, we see decreases in a lot of zooplankton taxa that are important food items. On the other hand, across much of the estuary, we see increases of invasive and small zooplankton taxa like H. longirostris and L. tetraspina. And in the South Central Delta, we'll talk on that in a second, we actually see increases of some other species too. So to try and wrap up some of these into conclusions and make some small predictions of what we could expect from zooplankton communities if we see increases in drought severity and duration. So first off, we can expect to see important zooplankton food for native fishes, such as the calanoid copepods we talked about, E. affinis and P. forbesi. We can expect to see those decline significantly, as well as the important Cladocera like Bosmina longirostris and Daphnia. We can really expect to see their populations decline, specifically in the important areas like Sassoon Bay and Sassoon Marsh. And these regions are very important, considered important nursery habitat to a lot of native fishes. So these important food items declining within those regions could have serious deleterious effects for a lot of our fish populations. Next up, we can also expect to see the community continue to be kind of dominated and saturated by more and more of these small invasive zooplankton species like Limnoithona tetraspina and the Mysid Hyperacanthomyces longirostris. And that could also have a compounding effect. So not only are there less and less of these important prey items that the fish have typically relied upon, but the ones that are coming in that are becoming more populous and more abundant in the area, they're a lot tinier. So each fish, when it's going around and looking for food to eat, is going to have to capture and spend more time feeding on these smaller zooplankton taxa because those larger taxa aren't as available as they used to be. So that takes more energy because you're having to eat more of them. And a side note, if you're interested more in the competition between the native mysids and our invasive mysids, my colleague Yugochi Akima has a poster on mysid fecundity that you can find during the poster session. I highly suggest you check it out. And also, looking at South Central Delta region, we can expect to see the predominance for zooplankton biomass throughout the estuary be concentrated in the South Central Delta during drought years. However, this region of the estuary can be really hostile to native fish, with typically some of the highest temperatures and some of the largest microsystems blooms that we see throughout the region. So there might be really good, a lot of abundant zooplankton in this region for fish to eat, but whether or not the fish can actually survive in that region to take advantage of that can be called into question. With that, that's kind of my predictions for the future of droughts and how they'll impact zooplankton based on what we've seen. And I'd like to open it up to questions. Thank you.
All right, it looks like we have a couple questions in the chat. So Wim asked, Arthur, have you tried taking salinity into account? In Sassoon Bay, you show decreases in biomass for those taxa that are most abundant in fresh water, such as P. forbesi and Cladocera. To me, it just looks like movement or contraction of their habitat.
Yeah, so that's definitely kind of the next step in this analysis, is moving beyond just, okay, drought versus wet years and trying to take a next step in looking at what are the actual driving factors driving the difference between these drought and wet years. So obviously a lot of that can be habitat opening up because of freshwater in Sassoon versus not freshwater in Sassoon, or community interactions and things like that. So that's definitely a next step going forward.
He also added residence time in the Delta. Calvin Lee asked, what particular aspects of the drought do you think is affecting the zooplankton community? For example, increased salinity inland, higher temperatures, changes in predator distributions.
I think all of those above that he just mentioned, that were just mentioned. So definitely salinity zone moving up and down through the estuary definitely kind of dictates where a lot of these zooplankton can live. Community interactions between predators like the invasive Ecartiella sinensis and its prey items, and then also higher temperatures could be favoring these smaller copepods. So definitely all three of those, and we're gonna have to examine those a lot further.
Okay, I don't see any other questions in the chat right now. I don't see any hands raised. We have time for another question if someone has any more questions. How much migration occurs?
Of individuals, I'm not really aware. I don't have an answer for that. But in terms of like the populations where they might end up given the flow conditions for a different year, you might get a lot of Pseudodaptomus up in the South Central Delta in a drought year, and then all the way down in Sassoon in a wet year. So there's a lot of variation on where those populations might end. Might be more highly concentrated, depending on things like flow and salinity. But in terms of an individual, I think Wim would probably have a much better idea of that than I would, in terms of like particle modeling and stuff like that. I think I see a hand from Wim.
Okay, I can't see it on my end, but oh yeah, go ahead, Wim.
Yeah, thanks. Yeah, thanks, Arthur. I don't mean to pick on you, but yeah, we have done quite a bit of work on vertical migration, because that's all they can do really. They're too small to really affect their own horizontal distribution except through their vertical. But I think I just want to sort of quibble about what I think is maybe the conceptual model in your head about how plankton work and what's in my head, which is that the plankton just go with the water. And that includes the larval fish and probably some of the juvenile fish as well. They're not that passive, but they're still moving with the water. And so when the water moves someplace, all the organisms move with it, the pelagic organisms. So instead of thinking of it as, well, opening up habitat, it's more like the habitat moved with the critters. It's a moving frame of reference that's kind of foreign to us landbound people.
Right. So I think this links into your downstream flushing paper, if that's right.
Somewhat, yeah.
Yeah. Okay.
Okay, well, thank you very much, Arthur. If you have any more questions, just contact Arthur. Our final presentation for this session is by Spencer Breining-Aday from CDFW, and he's going to talk about mysids and amphipods.
Hello, my name is Spencer Breining-Aday, and I work for the California Department of Fish and Wildlife. Today I'll be talking about the macroinvertebrates of fall, 9 years of pelagic mysid and amphipod monitoring in the upper San Francisco estuary. Mysids are shrimp-like crustaceans that inhabit the open water of the estuary and serve as an important food source for planktivorous fishes, including delta and longfin smelt. Monitoring for mysids was added to the Fall Midwater Trawl survey in 2011 in response to evidence that decreasing food availability was a factor in the observed pelagic organism decline. This monitoring was paired with existing fish and environmental condition sampling. The amphipods in these samples began to be recorded in 2013 to better understand the broader macroinvertebrate community. Data from this project has been used to inform aspects of the delta smelt resiliency strategy, including the North Delta Food Web Managed Flow actions. This community forms an important part of the food web of the system, and its dynamics may have a powerful influence on higher trophic levels. Samples are collected at 32 Fall Midwater trawl stations at the beginning of each month from September to December. A sled, like the one pictured here, is towed for 10 minutes in a stepwise oblique fashion to sample the full water column. The mysid net positioned at the center of the sled is made of 500 micron mesh and is designed to sample macro-sized organisms, or those typically larger than 1 millimeter in length. Pilot studies were conducted as early as 2008, but routine sampling did not occur consistently until 2011. Though the project is ongoing, for the purposes of this analysis, we will be looking at data from 2011 to 2019. In 2013, the amphipods in the sample began to be counted along with the mysids. Environmental conditions such as temperature, conductance as a proxy for salinity, and turbidity are recorded at the time of sample collection. Samples are taken to CDFW's Stockton laboratories, where the organisms are identified and enumerated. Length, sex, life stage, and fecundity data for these organisms is also recorded. Catch per unit effort, or CPUE, is calculated for each taxon at each station by dividing the catch at that station by the volume of water sampled. Annual regional catch indices for each taxon were calculated by averaging the CPUE of all stations in that region in that year. The unit for CPUE is organisms per cubic meter of water. The 32 stations sampled are divided into 8 regions for the sake of this analysis. Note that the Sassoon Bay region encompasses samples from Sassoon, Honker, and Grizzly Bays, as well as Montezuma Slough. Also note, Fall Midwater Trawl is the only study to routinely monitor open water macroinvertebrates upstream of the city of Rio Vista. Consequently, this remains an understudied habitat despite its importance for native fish. Let's take a look at the mysid community composition as told by our samples. Hyperacanthomyces longirostris is by far the most abundant species in the upper San Francisco estuary. An invasive species, it was introduced in 1993 and has subsequently taken over. In second place of overall abundance, Neomyces cadiocensis is the most common native species caught. The 5 remaining mysid species are caught only rarely. They are, in descending order of abundance, Orientomyces aspera, Alienacanthomyces macropsis, Neomyces mercedes, Orientomyces wanhiensis, and the elusive Deltamyces holmquiste. Neomyces mercedes was historically dominant in the low salinity zone and at one time served a crucial role in the diets of many fish. It is now only rarely seen in open water sampling. Now let's see how the Mysid community is distributed across regions. Here you can see the catch indices plotted over the years for each region. All regions have the same y-axis scale for ease of comparison. As you can see by all the green, Hyperacanthomyces longirostris is prevalent in every region. The highest abundance of mysids overall is in the Sacramento deep water shipping channel. The highest diversity, however, is in the two higher salinity habitats of Sassoon Bay and Carquinez Strait. Rather strikingly, Cache Slough and the lower San Joaquin River have almost no catch. The South Delta is also relatively devoid of mysids. If we take a closer look at Cache Slough and the lower San Joaquin, we can shrink down the scale by a factor of 100 to reveal that there is some catch of mysids here. They too are dominated by H. longirostris, but is simply on a much, much smaller scale, 2 orders of magnitude smaller. We'll discuss what this might mean later on. We may be able to explain some of the patterns we see across the regions by looking at how the different mysid species are distributed across the ranges of environmental conditions sampled. Starting with salinity, measured here in parts per thousand, we see the average CPUE of Hyperacanthomyces longirostris in each of a series of salinity ranges from freshest on the left to saltiest on the right. What is immediately apparent is that H. longirostris is abundant across all salinities sampled in this study. Projecting Neomysis cadiocensis in a similar way, we also see a similarly broad distribution. However, it is notable that Neomysis cadiocensis is not found in freshwater samples, in this case, those from 0 to 1 parts per thousand. Orientomyces aspera seems to be even more limited, having only been found in the higher salinity ranges. Neomyces mercedes is also broadly distributed across salinities, but it is markedly most common in fresh water. We can project species distributions across temperature ranges in a similar way. There is evidence to suggest that tolerance to higher temperatures may confer a competitive advantage to H. longirostris. Please check out Michelle Avila and Rosemary Hartman's 2020 paper on the subject for more detail. Even though our samples are collected in the fall, and the warmest water temperatures are most likely to occur in the summer, we can see some signs here of the importance of temperature tolerance in predicting species distribution. Hyperacanthomyces longirostris is the only species to be found at the highest temperature ranges sampled, in this case 25 to 26 degrees C. It is also interesting to note that Neomysis cadiocensis is found at higher temperatures in these samples than Neomysis mercedes. Could this explain the persistence of Neomysis cadiocensis despite the precipitous decline of its congeneric? I ask you now to pivot your attention to amphipods. Let's start by looking at the overall composition in our samples. Gammarus dibri and members of the genus Hyalella are the two most abundant taxa. Both have a predominantly epigeo lifestyle, meaning that they reside on surfaces, such as the stalks of vegetation. A relatively diverse collection of other species makes up the remaining portion of the catch, listed now in decreasing order of abundance. Carophium alienense, Ampelisca abdita, Americarophium spinacorn, members of the genus Crangonix, Americarophium stimsoni, Grandigirella japonica, as well as a suite of other taxa not explicitly identified in our lab. Looking at the geographic distribution of amphipods, here you can see the catch indices plotted over the years for each region. Again, all regions have the same y-axis scale for ease of comparison. The Sacramento River boasts the highest abundance of amphipods. Quite the opposite of what we saw with the mysids, the Sacramento deep water shipping channel has some of the lowest index values in the estuary. Though the dominant species Gammarus dibri is pretty widespread, the higher salinity regions of Sassoon Bay and Carquinez Strait support a completely different community's assemblage. Let's take a closer look at the two saltier regions. The amphipod community here is dominated by Corophium alienense and Amphipod abdita. These species are not commonly found in any other region. There is also a greater diversity of amphipod taxa here than there is in other parts of the upper estuary. It is interesting to note that the presence of Gammarus diberi seems to have increased in Sassoon Bay in 2017 and onward. In the last 3 years, it has been the most common species there. We may be able to explain some of these patterns in regional distribution by looking at the distribution of taxa according to environmental factors, as we did with mysids, again starting with salinity. Here you can see the average CPUE of Gammarus diberi plotted over ranges of salinities in parts per thousand, with freshest on the left and saltiest on the right. Gammarus diberi is prevalent at the lowest salinities. Looking at that same projection for Corophium alienense, its distribution seems centered on the middle range salinities. Looking at it for Amphipod abdita, it's found to be most common at higher salinities and is likely distributed into saltier waters downstream of the study area. For members of the genus Hyalella, they're most commonly found in fresh water, that is 0 to 1 parts per thousand. As you may recall from earlier, Cache Slough and the lower San Joaquin River were both almost devoid of mysids. But as you can see with these projections, they have a good number of amphipods. Let's take a closer look at these two regions. You'll notice off the bat that Gammarus diberi and Hyalella species are the most common taxa in this area. Both of these taxa have a predominantly epigeal life cycle. These areas also boast the highest abundance of Hyalella species in the upper estuary. There's reason to believe that they may even be increasingly prevalent in Cache Slough. These areas are also notable for their presence of submerged aquatic vegetation, SAV. Environmental conditions may provide a clue as to what is happening in these habitats. We can examine the distribution of species as it relates to ranges of turbidity in the same way that we have with temperature and salinity. Here you can see the average CPUEs for the two most common mysid species over a range of turbidities in NTU. The left side of the x-axis represents clearer water and the right side more turbid. Comparing the mysids to the two most common amphipod taxa, you'll notice that both mysids see a drop in abundance at the lowest turbidity range. Strikingly, Hyalella species are most abundant at this same range. This difference in distribution may have a number of explanations. The epigeal lifestyle of the amphipods as compared to the pelagic and planktonic mysids is likely a large part of that explanation. It is also possible that mysids may be missing from samples from the lowest turbidity waters as a result of vertical migration into the benthos that they may undertake in response to higher levels of light. The persistence of amphipods in regions relatively devoid of mysids may suggest a larger shift in the local food web. Speaking generally, mysids rely on planktonic sources of food, namely phytoplankton and other smaller zooplankton. Epigeal amphipods, on the other hand, like Hyalella species, eat the algae and other microbiota that grow on vegetation, and sometimes the vegetation itself, so they rely on SAV for food. Persistence of amphipods despite the extirpation of mysids could suggest a shift away from phytoplankton and towards SAV as the base of the food web in Cache Slough and the lower San Joaquin River. What does this mean for the fish that historically relied on the planktonic food web? I will leave you all with a parting question. Is the main stem of the Sacramento River moving towards the same community that we now see in Cache Slough and the lower San Joaquin? The bar chart shows the sum CPUE of all mysids and the sum CPUE of all amphipods at each station in the Sacramento River region in each year. As you can see from the map, the stations are arranged on the x-axis with the most downstream stations on the left and the furthest upstream stations on the right. Stations further upstream and closer to Cache Slough have lower counts of mysids than amphipods. In more recent years, that has been true for more of the stations, and for the last 3 years, every station has had more amphipods than mysids. And finally, all the data I used in this presentation is publicly available. You can find us on EDI at the following link or at the CDFW website. Thank you all for listening in, and I hope you enjoy the rest of the workshop.
All right, thank you for that presentation, Spencer. Arthur, I see that your hand is up, so go ahead and ask your question.
Yes, thanks. I apologize if we talked about this in the past. I can't remember. Does fall midwater trawl track when your mysid net gets full up with like SAV? And
yeah, go ahead.
Yeah, sorry. And if so, have you seen a correlation with like higher amphipod counts? Because I know we've seen that in the zooplankton trawls that we do. So I'm curious if you all see the same.
So we have commenting that will happen in the field when there's a lot of veg in the sled, but it's inconsistent. Inconsistent to the point where I haven't drawn a conclusion based off of that yet. That being said, though, we did add to our lab processes in this last year. So for 2020 and now 2021, we now record the amount of veg that is actually present in the samples when they come back to the lab. Based off like a visual inspection of it. And so that might actually be able to like show that sort of correlation that you found in your same EMP samples.
Interesting, thanks.
Yeah.
Okay, I don't see any questions in the Q&A box right now, but I did like Arthur's comment about the vegetation in the sample because that's something that we document for our study, and it's definitely interesting to see the differences when there's a bunch of veg in the sample versus not.
Yeah.
And Arthur has his hand up again.
Yeah, sorry. I just figured if there's no other questions in the Q&A, I'll keep asking questions. And I don't know if I want to spoil Christina's talk because I haven't seen it yet, but is there any examination of that switch? You talked, I got really excited about your mentioning like a trophic switch implication between mysids feeding on pelagic foodstuffs and amphipods feeding on benthic like SAV. Is there any indication or examination of that bearing out in terms of fish diets and like fish switching over from mysids to amphipods? And should I just wait and see Christina's talk?
That is a better question to ask Christina.
Okay.
Fish do eat amphipods. They do come up in the diets. I couldn't answer whether or not there's been a change in those regions. Like a lot of what I looked at, it seemed like the shift had already happened by the time our sampling started in 2011, because those regions were already dominated by mysids going back to dominated by amphipods going back to the point when we first started recording amphipods. So it's hard to say, like, I don't have the evidence to say this is when a shift happened and here's how we can see it in the fish diets. That being said, we do obviously pair our mysid and amphipod sampling with fish sampling with the fall midwater trawl, and the region where we find the most mysids is also the region where we find the most like planktivorous fishes, that being the Sacramento deep water shipping channel. That's the refuge for delta smelt. That's also where we find the highest amounts of the shad species we come across. So I think it's likely that it's not a coincidence that the places where we're finding fish are also the places where we're finding planktivorous fish is the places where we still see a lot of mysids.
All right, Calvin Lee wants to know if there's any aerial tracking of SAV.
That is a good question. I do not know, but if there is some, I would want to look at it. So if anyone can answer that, I would be really interested in seeing that kind of data.
Right, and April Hennessy has a comment. She said Lee Miller started seeing amphipods in striped bass stomachs in the 90s. That's when
wow.
EMP started recording them.
Christina has her hand up in the audience as well.
Okay, yeah, sorry, I didn't see it, but go ahead.
Arthur, to kind of add to what Spencer said, and it's not really ruining my talk because my talk is on larval fish, so they aren't really consuming mysids and amphipods at that stage,
Good.
at least when they're that small. But I can say that we have seen fish eating more on amphipods in recent decades, probably like in the last 10 years or so. I don't really know if we can say that there's been a shift with like the data that we collect and like the sampling design. I don't know if we can truly say that there's been a shift, but and especially because fish will eat mysids when available. So it's like, I don't know if they're truly shifting or if they're just like eating the macrozooplankton that they can.
Okay, we don't have any more time for questions, but someone pointed out that you could talk to Shruti at CDFW for some aquatic vegetation imagery.
That's very interesting. Thank you for that. I appreciate that comment.